Usually when we talk about a medical diagnosis, there's this expectation of precision.
Right, like engineering.
Yeah, exactly. You break your arm, the x-ray shows that jagged white line, and the doctor just points and says, there it is. It's binary, broken or not broken.
It's clean.
It is.
Yeah.
And honestly, it's comforting. We really like things to be visible. But then, well, you look at a patient who is struggling to breathe or someone with a really rare genetic disorder, and suddenly that x-ray machine is just entirely useless.
Yeah, because we're looking at a diagnostic landscape that is completely invisible to the naked eye.
Right. To figure out what's wrong, you have to zoom in way past the anatomy and step into the underlying chemical reactions that actually make life possible, which is exactly what we were doing today in this deep dive.
Where indeed.
We're using the 32nd edition of Harper's Illustrated Biochemistry as our source material today.
And I got to say, it is the absolute perfect tool for that journey. I mean, this text is a giant in the field. It's been continually updated for 83 years now.
Wow, 83 years.
Yeah, since its first edition by Dr. Harold Harper back in 1939. So the core mission of this book, and our mission for you today, is to provide a comprehensive, scientifically clear summary of the principles of biochemistry.
Right, and to show exactly how they apply to clinical medicine.
Exactly. Whether you are a medical student prepping for boards, a young professional in the health sciences, or honestly, just someone who is insanely curious about how your own body works, we're extracting the absolute core principles of this text for you.
And the central thesis of the book is really what grabbed me first. It argues that medicine and biochemistry are not like separate disciplines. They operate on a two-way street.
A very busy two-way street. Yeah.
Right. So studying biochemistry explains why a patient is sick, but studying sick patients actively reveals the hidden molecular machinery of human life. Let's unpack this for a second. It's kind of like reverse engineering a crashed car to understand how the engine was supposed to work.
That's a great way to look at it.
We only realize what a specific gear does when it actually breaks.
And that historical relationship, it's the bedrock of modern medicine. For a really long time, people thought of biochemistry as just this abstract language of molecules happening in a sterile laboratory.
Just test tubes and beakers?
Right. But it was the clinical observation of pathology of human disease that drove the biggest biochemical discoveries. The text actually gives us a beautiful historical example of this with an English physician named Archibald Garrod in the early 1900s.
Oh, right.
Garrod was studying patients with relatively rare conditions, most notably alkaptonuria.
That's the condition where a patient's urine turns dark upon exposure to air, isn't it?
Yes, exactly.
Yeah.
And Garrod didn't just document the symptom, he investigated the mechanism. He realized that these patients were actually missing a specific enzyme.
So to use the car analogy, a specific worker on the molecular assembly line was just missing.
Precisely. Because that enzyme wasn't there to process a chemical called homogenesic acid, the entire assembly line halted. That specific acid built up in the body, spilled over into the urine, and then when it hit the oxygen in the air, it oxidized and literally turned black.
Wow. So by analyzing the breakdown, the disease itself, he essentially proved that metabolism happens in these sequential steps.
Yes, and even more importantly, that genetics control those steps. He coined the phrase inborn errors of metabolism and essentially founded the entire field of human biochemical genetics.
Just from observing that one missing gear.
Right. He showed us that genetics and metabolism are intrinsically linked. And you see this pattern everywhere in the text. Take sickle cell disease, for example.
Oh, yeah, that's a classic one.
By investigating why patients with sickle cell had those misshapen red blood cells, scientists discovered that a difference in just a single amino acid in the sequence of hemoglobin alters the entire structure and function of that protein.
It's crazy when you think about it. A single microscopic typo completely changes how the protein folds.
Exactly. And that didn't just explain sickle cell. It taught us the fundamental rules about how proteins fold and function in every living thing on Earth. The same goes for familial hypercholesterolemia.
Which is a genetic condition causing severe heart disease at a really young age, right?
Yes, very early onset atherosclerosis. Studying that disease didn't just explain why those specific patients had high cholesterol. It unlocked a much broader understanding of how cell receptors actually work.
Oh, wow.
Yeah, it mapped out the complex mechanisms of how molecules are taken up and transported across all cell membranes.
Even cancer research fits this pattern, doesn't it? The text notes that studying oncogenes and tumor suppressor genes in cancer cells is what originally taught us how normal cell growth is controlled.
Yes, exactly. We literally had to see cell division spiral completely out of control to map the brakes and accelerators that keep healthy cells in check.
So for you listening right now, this is why this context really matters. Knowledge is always most valuable when it's applied.
Absolutely.
Seeing disease not as some mysterious curse, but as a specific breakdown of normal biochemistry, it just removes the mystery. It replaces fear with molecular logic.
And allows a clinician to pinpoint the exact site of the intervention.
Okay, so if we know how the machine breaks, how does the machine actually run? Let's move from the philosophy of this two-way street into the physical hardware of the body. The first half of Harper's biochemistry, so that's sections 1 through 6, is all about how we are built and how we are fueled.
Right, so we're looking at the structure and function of proteins and enzymes, bioenergetics, and the metabolism of carbohydrates, lipids, and amino acids. This is the foundation of the house.
And I noticed the 32nd edition has some really specific updates here, particularly in Chapter 9, where they expanded the coverage of zymogen activation in enzyme regulation.
Yes, that's a great section.
From what I gather, a zymogen is basically like an inactive precursor of an enzyme. The body produces an enzyme in a safe, dormant state, and it only gets activated when and where it's actually needed.
It is a brilliant evolutionary safety mechanism. I mean, think about your digestive enzymes. Their entire job is to aggressively break down proteins.
Right.
You do not want those enzymes actively breaking down proteins while they are still sitting inside the delicate cells of your pancreas where they're made.
Oh, that would be bad.
Very bad. They need to stay inactive as zymogens until they safely reach the digestive tract.
It's like shipping a chainsaw with the spark plug removed so it doesn't accidentally turn on in the delivery truck. The body just chops off a little piece of the zymogen once it reaches the stomach, and boom, the chainsaw's running.
Okay, that is a very visceral way to picture it, but mechanically it's very accurate. And to keep those molecular chainsaws running, the text spends a great deal of time on bioenergetics.
Right, the fuel.
Exactly.
Yeah.
It details the role of high-energy phosphates, specifically ATP, or adenosine triphosphate. ATP is the energy currency of the cell. The book meticulously details how energy is captured from the food we eat and transferred via the respiratory chain in a process called oxidative phosphorylation.
Okay, stop right there. Oxidative phosphorylation is one of those textbook terms that immediately makes my eyes glaze over.
Fair enough.
Walk me through what that actually means mechanically. Like, how does a piece of bread turn into ATP?
Let's break it down. When you break down food, your body strips high-energy electrons from that food. Inside your cells, specifically in the mitochondria, those electrons are passed down a literal chain of proteins, the respiratory chain, like a microscopic game of hot potato.
Okay, hot potato with electrons.
Right. And as the electrons are passed along, their energy is used to pump protons across a membrane.
So we are building up pressure, like pumping water behind a dam.
That is the perfect visualization.
Yeah.
You build up this massive chemical pressure of protons on one side of the membrane. Oxidative phosphorylation is simply the process of opening the floodgates.
Oh, I see.
The protons rush back through a specialized molecular turbine called ATP synthase. The physical spinning of that turbine forces molecules together to create ATP.
Wait, really? We are essentially eating food just to build up chemical water pressure behind a microscopic dam.
That's exactly what's happening.
That is incredible. And the text mentions Louis Pasteur figured out the early stages of this without even knowing about mitochondria. We all know Pasteur for pasteurization, obviously, but he discovered that cell-free yeast preparations could convert sugars into ethanol and carbon dioxide.
Right, he was witnessing fermentation, which is
Yeah.
basically glycolysis, the early breakdown of sugar.
And he proved that you didn't need a whole living organism to perform these complex chemical reactions. You just needed the enzymes extracted from them.
Which was a massive paradigm shift.
Yeah.
It proved biological processes are chemical reactions that can be isolated and mapped, and the text maps them with incredible granularity.
Yeah, it really gets into the weeds.
It does. For instance, when it covers the metabolism of lipids, it details how fatty acids that contain one or more carbon double bonds are categorized as polyunsaturated.
Right, we see polyunsaturated fats on nutrition labels all the time, but the book shows you the actual structural kink in the molecule that the double bond creates.
Exactly. And it also dives into incredibly specific intermediate molecules. It mentions how delta-1 purulane 5 carboxylate acts as an intermediate in the metabolism of the amino acid L-proline.
Let's pause on that word intermediate because it is crucial for anyone studying this text. Metabolism is not like a single leap from raw material A to final product Z.
No, not at all.
It is a staircase. You have to convert A to B, B to C, C to D. Molecules like that delta-1 pyrroline, whatever, are those stepping stones. Highlighting them shows the sheer depth of the chemical pathways Harper's covers.
But this raises a huge problem for anyone trying to learn this material.
Yeah, tell me about it.
When you open a textbook like this, you see hundreds of these complex pathways. You see the glycolysis pathway, the lipid oxidation pathway, the amino acid degradation pathways. Is there a simpler way to view all this? Do lipids, carbs, and proteins act like isolated factory assembly lines? Or are they a single interconnected web?
And that is the most crucial conceptual hurdle for any student of biochemistry. It is absolutely a single, highly interconnected web. While we study them in isolated chapters for the sake of our own sanity in the human body, all these distinct pathways are intimately linked.
They share those intermediate stepping stones.
Meaning the breakdown product of a carbohydrate might actually be used to synthesize a lipid.
Yes. Or an amino acid might be converted into glucose if the brain desperately needs energy. They are all ultimately feeding into the exact same goal, maintaining cellular homeostasis and energy balance.
And this is exactly why a defect in one single pathway can cascade into a massive systemic issue.
Give me an example of that cascade. If one gear breaks, how does the whole car crash?
Well, let's look at pyruvate. Pyruvate is a key crossroad molecule, an intermediate in the metabolism of carbohydrates. Normally, it gets oxidized and enters that mitochondrial dam system we talked about to produce a massive amount of ATP.
Right, the normal fuel path.
But if there's a defect in pyruvate oxidation, say, due to an enzyme deficiency or maybe a lack of oxygen in the tissue that pyruvate has to go somewhere else, it gets shunted into an alternative pathway and becomes lactic acid.
Wait, so the body is essentially forced to dump this toxic byproduct just to keep the lights on?
Exactly. And if that lactic acid builds up in the bloodstream, the blood's pH drops, you get lactic acidosis. A single blocked chemical reaction in the cells cascades into a life-threatening systemic crisis. That's the interconnected web in action.
Which brings us to a really fascinating transition. If the proteins and the energy pathways are the hardware and the fuel of the cell, the second half of the textbook, so sections 7 through 11, moves into the software and the communication systems.
Right. We dive into the replication of informational macromolecules.
Let's ground that term for a second.
Yeah.
Informational macromolecules is a very clinical way of saying the blueprints. We are talking about DNA, RNA, and the genetic code.
How information is stored, copied, and translated into the physical reality of the body.
Yeah, it's the transition from hardware to information. And beyond the software, it's about how the cells actually talk to each other.
Because if DNA is the hard drive, then hormones and cell membranes are basically the Wi-Fi network. They allow these trillions of individual microscopic cellular computers to coordinate, to share information, and to act together as one single human being.
The clinical term for that Wi-Fi connection is signal transduction. A hormone docks to a receptor on the outside of a cell membrane, and that single event triggers a whole cascade of chemical changes inside the cell.
Like turning genes on or off, or telling the cell to release a specific molecule.
Exactly. It alters its entire behavior. And the 32nd edition actually reorganized and expanded on one of the most elegant examples of this kind of molecular communication, the Bohr effect.
The Bohr effect. This is a beautiful piece of physiological chemistry. It describes how carbon dioxide and hydrogen ions affect hemoglobin's affinity for oxygen.
Essentially, it explains how hemoglobin knows exactly where to drop off oxygen.
And I've always struggled to visualize this. Because if hemoglobin is picking up oxygen in the lungs and holding on to it tightly, how does it suddenly, know to let go when it reaches a flexing bicep?
Think of hemoglobin like a tightly clenched fist.
Okay, a fist.
And the oxygen is a sponge held inside that fist. When the fist is clenched, the sponge goes nowhere. Now, an actively flexing bicep is burning through energy, and as a byproduct of that metabolism, it produces carbon dioxide and protons.
Which creates an acidic environment.
Exactly.
So the working muscle is basically sweating acid into the bloodstream.
That is a brilliant way to phrase it, yeah. And the Bohr effect dictates that when the hemoglobin fist floats into that acidic CO2-rich environment, the acid acts as a chemical trigger. It interacts with the amino acids in the hemoglobin, subtly shifting its entire molecular structure.
Oh, wow.
The acid literally forces the fist to relax.
So it drops the sponge exactly where it's needed most.
Yes. It automatically releases its oxygen payload, purely based on the local chemical environment. Then it binds to that CO2 and carries it back to exactly where it needs to go, to the lungs.
That's amazing. And the textbook actually uses this mechanism in its exam questions to test clinical application. You pointed out a brilliant question in the answer bank earlier about hyperventilation.
Oh, yes.
Walk me through the mechanics of that. Why does rapid, shallow breathing hyperventilation lead to an increase in blood pH?
This is a classic board question, and it's pure biochemistry applied to the clinic.
Yeah.
Let's trace the domino effect slowly. Domino 1, a patient starts hyperventilating. They are exhaling carbon dioxide from their lungs much faster than their tissues are producing it.
Okay, and domino 2, because CO2 combines with water in the blood to form carbonic acid, blowing off all that CO2 means you're rapidly losing acid from your bloodstream.
Right. Domino 3, because you are losing acid, the blood becomes more alkaline. The pH increases. This state is called respiratory alkalosis.
And here is where it gets crazy. Domino 4, that alkaline blood, changes how proteins hold on to minerals. Specifically, calcium starts binding much more tightly to proteins in the blood.
Which leads to domino 5. A sudden drop in free-floating calcium in the bloodstream causes nerves to misfire. That misfiring is what causes the classic tingling in the fingers and lips that people get when they panic and hyperventilate.
You start with fast breathing, and you end up with tingling fingers, and every single step in between is just a chemical equation balancing itself out. It all comes back to the molecules.
But we also have to ask what happens when the Wi-Fi goes down. A failure in signal transduction, or a breakdown in these communication pathways, is exactly what leads to the clinical pathologies covered in the final sections of the book.
You mean things like cancer and aging?
Precisely. Cancer is fundamentally a disease of cellular communication. A cell stops listening to the signals telling it to stop growing. Or, a cell receptor becomes permanently stuck in the on position, constantly telling the nucleus to divide. Thrombosis, or inappropriate blood clotting, is another example. The intricate signaling cascade that tells platelets to aggregate happens at the wrong time or in the wrong place. These aren't just gross anatomical failures, they are molecular miscommunications.
So as we zoom back out, what is the ultimate takeaway for you, the listener, from this massive text? The core message is that biochemistry isn't just an abstract language of confusing diagrams and chemical structure.
No, not at all.
It is the fundamental vocabulary of life, health, and clinical medicine. Whether you are prepping for a high-stakes medical exam or you're just incredibly curious about your own biology, mastering these chemical reactions isn't about rote memorization. It's the key to truly understanding human biology.
And as an educational content creator, my goal is to make sure this actually sticks with you. Knowledge only becomes yours when you force your brain to actively recall it. I want to leave you with a specific review exercise to reinforce what we've unpacked today.
Okay, hit us with it.
Before you move on with your day, maybe while you're driving or making a coffee, take a moment to explain out loud to yourself, to mentally trace a clinical symptom back to a biochemical concept we discussed.
Oh, that's good.
Yeah, walk through the relationship between exhaling CO2 and the loss of carbonic acid that leads to tingling fingers. Or explain to yourself how a single amino acid substitution alters a red blood cell in sickle cell disease. Don't just think it, speak it. If you can explain the why out loud, you truly own that knowledge.
If you can teach it to an imaginary patient in the room with you, you've mastered it.
Exactly.
As we wrap up this deep dive, I want to leave you with a provocative thought that builds on what we've discussed. We talked extensively today about the two-way street, how studying sick patients reveals the hidden biochemistry of human life. But the text also touches on the Human Genome Project and the incredible advances in genetic mapping.
The mapping of our informational macromolecules is advancing at a staggering pace.
So here's the question for you to mull over. As our genetic and biochemical mapping becomes near perfect, will that two-way street eventually become a one-way street?
Oh, interesting.
Will we reach a point in the near future where we no longer need to study sick patients to understand biology because we can predict every disease, every metabolic bottleneck, purely from an individual's biochemical code before symptoms ever arise?
That is the ultimate promise of precision medicine. It changes the entire paradigm of what it means to diagnose a patient.
Right. Will the X-ray of the future just be a biochemical sequence? When you think about it, that broken bone we talked about at the very beginning, maybe the jagged white line on the film won't be the diagnosis anymore.
Yeah.
Maybe the real diagnosis will be the invisible molecular code that predicted the bone's density would fail 10 years before the fall ever happened. Something to think about as you navigate the invisible chemical world inside you. Keep asking why, keep looking deeper, and we'll catch you on the next one.